All right, we're back for part three. I guess I didn't beat you up enough with part one and two. This will complete chapter six as we move forward. This is a easier part of this particular chapter from the standpoint. Concepts are straightforward, but these are ones that we do need to go over. They are very important in terms of disease production and what to expect as a clinical microbiologist as we work with these microorganisms. And so we're going to be looking at cell division, doubling time. It's amazing. I mean, you think about it, some, not all, some bacteria can double every 20 minutes. Now think about that next time you leave a potato salad out on a table while you're going in church or some function or something, and you come back out and then you wonder why you have severe profuse diarrhea 4 or 5 hours later. Staph will grow and start producing preformed toxins and things. Well, they're toxic to us, it's just their waste material. They don't have a toilet to flush or anything. And you eat it, and then 4 to 6 hours staph, 6 to 8 hours salmonella, those sorts of things. And I know what I'm talking about when it comes to diarrhea because I have a thesis on feces. Yes, I do. I have a master's, and I presented a paper at the International Diarrhea Conference. I tell you, there was a lot of, well, anyhow, we'll move on from there. Binary fission. One cell becomes two. So there is no male, female, there's no whatever. Binary fission means that at some point in time, the signaling internal to the cell, it divides. One cell gets one copy of the chromosome, which is attached to the membrane, and then the other cell gets the other chromosome that's been copied, that's attached to the membrane. There is no mistake. Each segment will get a new or the chromosome, and the new cells are off on their way. Duplicate meaning cloned, absolute clones of each other, so they're identical. Um, the septum forms between the cells, as we'll see here. So here's sort of a diagram. Here's a cell. There's a signal that's going on. It needs to begin the division process, which is chromosome controls, and then copies are made. Then it's not shown here, but they actually adhere to the membrane that keeps in the two, and then a septum forms, and boom, they divide. Pretty straightforward. Generation time or doubling time. Please know these words. Highlight that, underline it. Generation time, doubling time. It's the time required for a complete fission cycle. In other words, to split. Now, if you have 10 to the 10th bacteria growing on a salad, and the doubling time, that means 10 to the 10th, well, each of the cells that are undergoing, which is the majority of them probably, will double, and so that's a considerable and large numbers. Now, we have to kind of plot this in a logarithmic mode because it doubles. And so when you have a logarithmic kind of growth pattern, we plot it on a logarithmic type paper or graph paper, and it shows a smooth sort of increase. If we did it as a straightforward plot, it's just too much data too quickly for it to be represented properly. So I point that out. You may see some plots here in a little bit. So let me show you some bacteria and average generation times. Usually we say 30 to 60 minutes. I've worked with microorganisms 20 minutes. Some of the shortest times would be 10 to 12. I haven't worked with those. Mycobacterium leprae, which has a generation time of 10. This causes leprosy. That's quite amazing. That's impressive. It takes 10 to 30 days. I mean, you think about that. Mycobacterium leprae. Anyhow, you get the point. 10 to 30 days. Environmental bacteria have generation times measured in months. Azotobacter violandi, I made reference to, that nodulates plants and things. Very slow grower. You got to be really patient to work with that organism. I did as an undergraduate. I learned a lot doing undergraduate research Wake Tech does support that. And if you're interested in a project, and I do some, but there are other instructors that do microbiology related undergraduate research. By all means, ask, inquire. We will get you set up, even if it's online for right now. That's fine. Eventually, it can't be forever, and we'll be back in our very nice building before long. Most pathogens have really short generation times, and oddly enough, I read a report where they took bacteria up into space in the space lab, and they found that certain strains like Streptococcus grew faster and were much more virulent in space. I found that interesting. I don't know what the implications or why that is, but I don't even know why I brought it up. But anyhow, you got the point. So here is the mathematics behind this, and again, I was kind of showing in the logarithmic, nice straight sort of curve, and if you do a flat plot, you can see where it's asymptotical. It just moves straight up, boom. And where in a log plot, it shows more gradual. This is how we're supposed to publish anytime in a professionally reviewed paper. We always use logarithmic. So if we start with 1, binary fission, then we get 2, and then 4, 8. The best way to calculate that is something called 2 factorial. And all you have to do is raise 2 to the power of however many generations that it's undergone, and it will tell you what the number of cells will be, which is a nice thing. I mean, it's predictable. It's one thing, sometimes bacteria can be very predictable in some ways. So the size of the population can be calculated by the time, whichever number you've started with, n, times 2 raised to the power of n. And so n represents the starting number of cells, exponent n denotes the generation number, and 2 times to the power of n represents the number of cells in that generation. Really straightforward. It is an equation that I do use in a microbiology lab, and we are going to be covering solutions and dilutions Dr. V's way. You had what I call the primer. If you're from Britain, how about primer if you're from here? That was in the chapter 2, I guess it was, or lab 2. You had to do some numbers, and now I'm going to actually apply some of that in solutions and dilutions in the week 5 lab. Population growth curves, that's what we're going to look at now. So we pretty much anytime I work with a pathogen, and especially a new one, I worked with Alkaligenes faecalis one time, knew nothing about it. Alkaligenes kind of tells you that it produces some basic waste product because it's alkaline, alkaligenes, and faecalis is found in the feces, of course. I always get stuck, never mind. Growth curves from those organisms let me know how I need to learn how to work with the organism, especially if I'm not familiar with it. Usually my first attack is to figure out what the minimal media is, the absolute defined media that's required, everything it needs to grow, that I know exactly what it is, and that way I can make mutations and do various things. It's a long story, but you get the gist of it. So to do a growth curve on one, you place a very small number of cells into a sterile liquid medium. It's usually some sort of media that they like, and it can be complex, no problem. I personally prefer minimal media because I'll report it as such, and that way I can control every parameter of growth, how much carbon source, all that. But that's not typically done. Just growing it in complex media would be sufficient. Like a brain heart infusion, very tasty. It's loved by many bacteria. Incubating the culture over a period of several hours, sampling the broth at regular intervals. I can't talk today. Regular intervals is really, really important. You want to take samples so you can plot it and have some sort of sense as to when you do this. Plating each sample into a solid media, and then you use a hockey stick, and I'll show you about that, and spread the bacteria. and across the plate so you can count them. Now, the problem with it, with just plating the way they've got this written here, it's not as easy as it sounds like. Because when the bacteria start to grow in a doubling time the way it is, every 20 minutes, you can imagine that before long it would be too numerous to count on a plate. So that's why a microbiologist has to do dilutions. And I'm going to show you how to do dilutions. It is very easy, but I don't know, it does involve math, and sometimes brains shut down. But the way I do this, you do not need to have second year of calculus or differential equations or any of that. In fact, you can even leave your shoes on, because the math that I do are simple enough to do with your hands if you do it that way. So idea is, I don't want you getting nervous about it at all. Now, you're going to be needing to do math in the future. So some of the things I'm going to be talking about when you get into more of the pharmaceutical aspects in nursing, they're going to make you go through a lot of different types of math, and you memorize formulas and do things. The way I'm going to show you is the way I was taught when I worked at Burr's Wellcome. It is how to think on your feet and to do the math correctly. And it's down to earth. You hit the ground running with this type math. And when I was there, I'll just share this with you. I was amazed. I said, why don't they teach this in school, to do the math this way? And they don't. And it makes sense from the standpoint, with the way you teach it in school. It is scientific, it's done with equations, and there's no deviations, and blah blah blah. If you understand how math works, which I will explain, it's really straightforward. You can figure it out on a backside of an envelope or in a lab situation, and you check your work, of course, but you'll come up with the same answer, and you can do it without taking a calculator and notebooks and everything with you. Sometimes you just have to do things on your feet. And again, you document if it's required as an SOP. When you're doing something, you follow the instructions. But if you're left to your own on the floor, I'm going to show you how to survive that and do it correctly. And we'll do that in solutions and dilutions in the lab of week 5. So sampling the broth at regular intervals is really important. Plating each sample onto a solid media so we know roughly with doing dilutions and the like. And then you count the colonies. And there's even tricks for doing this. When we count the colonies, you don't have to count them all. What you do is you have a counter that divvies up the plate in sectors, and they're all equally drawn. So all you have to do is, let's say, count out one of the 4 sectors, then multiply what you found by 4, and you're on your way. You know exactly how many there are. And so there's tricks to the trade. One of my favorite sayings, you'll hear me say it a lot, try to work smarter, not harder. And that just means thinking about what you do. That's why we do pre-labs. Or at least have you think about the lab before you do it. That way you can think of a smarter way to do it. You don't have to do it exactly, in terms of the order and all that, as it says in the manual. In fact, we're going to be deviating because I want you to do some of these things as a microbiologist would do them, because that's, hey, the course. I noticed the segments are really kind of genericized, which is fine. I mean, it's all good. The equipment's there, and I'm excited because we'll get to work with some of these things, and we'll do it together. All right. So steps in a viable plate count. So you inoculate, you plate out, and then, however, they have it set for every 60 minutes. They take samples, and you can see when they plate them out, and you get down here, it's like TNC. Now, up to about, I would say about here, we start to see problems with too many colonies to count. Usually my break off is somewhere between 45 and 80 colonies on a plate, depending on how big the colonies are. There's a lot of factors, but I usually start doing within maybe an hour or so. I start doing dilutions, and you can make it apparent when you start you can put these in the refrigerator or whatever as you do these, or you can plate them directly. If you plate them directly, then you have to do some dilutions, and you don't know the numbers, and so sometimes you do the solutions and dilutions without really knowing it, and so you get really good at them. And let me tell you, I can't tell you how many dilutions I have run in my life. It is it's, I don't know, it would be kind of like, it shows you how old I am. So we represent the data, if you look at it, linear and then logarithmic. And we frown upon using linear data when we have logarithmically growing bacteria. In other words, they double, and they do it in a function of 2 raised to a power. So we want to plot it on logarithmic paper. And what that means, if you haven't used logs, you notice that each division goes up by a factor of 10. And here we're just kind of going up depending on how we represent the data. And the problem becomes that here you really can't get good demarcation of what the numbers are. I mean, tell me what that number is. I mean, that's almost impossible. It'd be total guesswork. Where in a logarithmic, it's 10, and usually these are even subdivided out that you could figure out that it was 15, or it's 52 or something like that. The way that the logs, it's asymmetric logs, so you get more and more spacing as it goes in a direction. So it makes it easier to actually pin down and looking at a graph. This makes it really hard. You know, the difference between this point 5,000 and 6,000. I mean, really, it just is not very accurate. And so you might see in a publication more like this. Here's the lines I'm talking about. You see the spacing increasing as it goes down. So we have 10 to the 7th, 10 to the 8th, and you can literally figure out that somewhere in between. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it's before 1 times 10 to point, let's see, that'd be 1, 2, 3, 4, 5, maybe 5 cells, so it'd be 5 times 10 to the 7th, and going on up. At least the guessing, if there is guessing to be done, is a lot more accurate using logarithmic paper, and that's why we use it. And we measure on a spectrophotometer. Once you've done your standards, you can see that the counts per CFUs per ml and the absorbance track each other. And so once you've done your standards, then you don't even have to plate anymore. You can just use your absorbance patterns. It's a logarithmic type of relationship. So yet another reason why we want to use logarithmic, because it really allows us to pinpoint numbers, and using the absorbance meter as spectrophotometer, it just makes life so much easier. And there is a slight disparity depending on, I've had some that are on top of each other. My guess is probably the effects of not doing enough for the standards or something. I don't know. But anyhow, it's not that much of an issue. The factor becomes it's off by time. And maybe it's just because the absorbance is just more accurate early on. I don't know. So I just want to, at the end, we have 1 times 2 times 10 to the 11th. Now, we didn't see 0.2 of a cell. Yeah, I always get that. Now, it was 12 on the plate. In order to make it in scientific notation, we take the 12 and we make it 1.2. So it was 12 times 10 to the 10th, and we just change it to 1.2 times 10 to the 11th. And just using the power of exponents. So that's what that means, and I don't want you to say, gee, Dr. V thinks that we can get 0.2 of a cell. Really? Geez, what's he smoking? No. Stages in the normal growth curve. We have a log phase, and I worked at Burrs Wellcome, and I was well aware of using fermenters and fermentation. And there's a phenomenon, it's sort of a lag phase. It's sort of like the bacteria are gearing up to grow, and no one's really knows what the cells are doing, but they're not doing a whole lot of anything that we can see. They're just not growing very much. It's a flat period of growth. They could be playing poker, they could be calling out for pizza. I don't know. your guess is as good as mine, but they're not multiplying. And so during this lag phase, the population of cells are sparse. And the idea is what I usually have done is I produce a pre-culture that already lags out, and then I inoculate a large volume, and the lag time is greatly shorter. So we have a bunch of cells geared up, ready to go in the media that they're used to growing in, and you're off to the races. Then the next stage we need to talk about is the exponential growth, and we call that logarithmic or log phase because it's growing geometrically. How many words to describe the kind of the same? Exponential, logarithmic, log, geometric. They all mean represent the same thing in slightly different. This is more of a mathematical. Exponential is exponential with 10 to the raised to the so and so. You see, so that's plotted in a logarithmic, a log base 10. So that's not to be confused, but these are the lingos that we're used in labs, and a lot of times in microbiology you see a logarithmic used a lot. So don't fear the word, it's really straightforward. And so bacteria will grow as long as they have an environment that they haven't used the bathroom too much, or if it hasn't been cleaned, waste products build up and it's toxic, and that's not good. And so usually on a sustained or a continual batch fermentation, we take out media, filter it, and add fresh media back. And so that way the bacteria are experiencing a somewhat favorable environment. It has adequate nutrients, and the waste products are being dealt with. So we move in little porta potties and that sort of thing. No, I'm just kidding. Joking, we don't put porta potties in there. Stationary growth phase, cell birth and cell death rates are equal. So we grow exponentially, and then we start to curve up, and that's kind of flat. And that's the stationary, where we have sort of the birth and cell death canceling each other. And that's usually because we're starting, the limiting factor is the nutrients and the oxygen in the media. Now, a continuous batch type of fermentation is that this is dealt with. But still, even on a continuous batch, you produce just enough to sustain the population, not too much more. It all depends on why you're growing the cells at such high numbers. And we have excretion of organic acids and pollutants. That's a nice way to put, bathroom excrement as pollutants into the growth media. That's a very nice way to put that, but really it's just bacteria using the bathroom. So anyhow, it goes in. So usually in a continuous batch we deal with it, but if you're just doing it in a closed system, you're not refreshing. This is the typical thing that happens, is that you go from a plateau and boom, goes down. We call the going down part the death phase. The cells begin to die at an exponential rate due to the buildup of waste. They're toxic, and it's really nasty. I mean, I would not. I could tell you a joke about a porta potty, but I won't. Mixed company. It's just no. I don't want to see myself on CNN or anything. Speed at which death occurs depends on the resistance of the species and how toxic the conditions are. Slower the exponential growth rate. Okay, of course. Viable non-culturable state. Now this has been around for a long time, and to be honest, when I first saw this, I said, what the heck are they talking about? So when a cell is viable, it's growing. Now you could, in theory, have a spectrophotometer and it has some density. You measure it and you say, okay, I got a certain number of live cells. That could be a total lie. None of those cells that are in the mass that's taking up density of that tube, in other words, it's cloudy, but really there's nothing viable. There's nothing living. Viable and living are the same thing. So viable cells, if they're not viable, they're dead. And so really a microbiologist wants to know, of those cells that are in that liquid, how many are actually alive? That's more important to me, because the live ones usually can cause problems. And so many cells in the culture and death phase stay alive. Now, one of the things that bacteria will do, they have no shame. Now, what did I say? No shame? They will stack other dead bacteria on top of them. just so that they protect themselves from the toxins from that waste product. So they kind of hunker down and they'll sit at the bottom. Bacteria, subject to gravity, and dead cells will end up floating to the bottom, and they just sit there. And some viable cells get covered, and it protects them. And so they kind of sit there and twiddle their thumbs and waiting for good times. They're hoping that they will have some change. So we'll not grow culture medium or miss the colony count. So a lot of times that happens, and so we want to know viable cell counts. So here's the plot. Here's the lag phase, growing exponentially until it starts to equal deaths and cell, and it's usually limited by the media components, and then the waste products start to build up. Or what was that wonderful term they used? What was it? Biochemical pollutants. Yes, as biochemical pollutants start to build up, we start to see this descent, death phase. And although some cells come up with creative ways, waiting for some good news, maybe. And there you go. That is the growth curve, and it's pretty standard stuff. Practical importance: the tendency of populations to exhibit phases of rapid growth, slow growth, and death has implications in microbial control, food microbiology, you can imagine, and culture technology, like batch fermentation. Unless you're aware of it and have been out to pharmaceutical companies, bioengineering or biotechnology has utilized bacteria as a little as a sort of factories that we genetically modify, and they create whatever we want. So understanding the culture technology. Now, when I say that, it's not social media where they're going on Bacteria book or Facebook or whatever the bacteria use for their social media. That's not part of their culture. What they're talking about is their growth rate and how they're vulnerable to various things. A person actively shedding bacteria in early and middle stages of infection are more likely to spread than a person in the latter stages, because there's so many dead, and the environment, the local environment, is not too conducive, so a lot of the cells are already being inhibited. Terms: turbidity. And the best way, if something is turbid, in other words, if it's clear, that's the kind of the antithesis, like water in a test tube, it's clear. But if you start to add orange juice to that or something else, Pepto Bismol, okay, it starts to get turbid. In other words, you can't see through it very well. And the more you can't see through it, the more turbid it is. And so we use turbidity in a spectrophotometer because of how much light is blocked by the stuff floating, okay? So greater the turbidity, the larger the population size. Calibrate growth using turbidity with viable cell counts. And I've already made reference to that. I've done these billions and billions of times, but I like how they did this. They put a ruler behind there to see that you can see the text, at least with a liquid. Refractive index has distorted the text because we already talked about refractive index, but I'm going to go over that again. And so low turbidity, it means higher amount of light can get transmitted. Okay, so there's usually two settings on a spectrophotometer: is absorbance and how much is transmitted. So here's the transmitted, and it's easy to calculate what the absorbance would be. Is if you start off with with something that has zero turbidity, that's your 100%, and then later on you measure it, you can subtract the percent transmitted at the very beginning from what is being transmitted later, and that's your absorbance, because that would be the remainder part. So anyhow, you would plot if it's transmitted, you report it as transmitted source. You can see, and it becomes turbid. Now that's a lot. That's yucky. That's pretty high. That's over pretty out here, so you can see it's pretty dense, and the amount of light being transmitted is pretty low. And so anyhow, there you go, and that's directly proportional to the number of cells. And if I've done a viable cell count to each of these readings, then I know roughly how many cells are there, and there you go. So it's just to show the same plot again. normally. So direct cell count measured microscopically. It's really easy. Coulter counter. And this is nice. The Coulter counters electronically scans the fluid as it passes through a tiny pipette, and that's I remember using Coulter counters. They're not that accurate, to be honest with you. Wellcome kind of frowned on using those. Flow cytometers, that's a different deal. It's similar to a Coulter counter, but the receptor, the light receptor cells, it wasn't diodes. I forget what it was. It was light absorbing diodes of some kind. Anyhow, anytime a cell would block it, even as small as bacteria, it could be detected. So flow cytometers work very well, and they can even, because of the sensitivity, even differentiate between live and dead cells. Now, a lot of times dead cells get distorted, you can imagine, because, well, that's what they do. And so a flow cytometer can do that. It can even not only count, but you can put a deviating nozzle on there so you can actually separate viable from non-viable and things like that. We can also use real-time PCR, polymerase chain reaction. If you remember, the function of PCR, just so if you're not aware, is just to amplify DNA. In other words, it does that at higher temperatures than we normally would think about replicating DNA. And its ability to quantify bacteria and other organisms present in the environmental or tissue. In other words, it can use PCR to detect if some sort of DNA that we're interested in, very small amounts, are present or not. And so genetic probing is a very sensitive tool if you want to detect very, very small amounts of bacteria. The direct count is easy to do because I've mentioned the grids, and a grid will have a certain number. It's known. It's usually printed right on the grid. And if you count one series of squares, let's just take this square, and you count one section of that grid. Let's say 1, 2, 3, 4. Okay, so there's 4 bacteria here times 4 times 4. That's 16. So there's 4, 5, 6, 7, 8, 9, 10, 11. Well, no, I counted down here. Anyhow, it's supposed to count within there. This is just a representation. But the idea is you can give a representation of how many are there. It's usually pretty accurate. This is a drawing, so it's probably just not real. Here's a Coulter particle counter. It's used a lot in food microbiology, just to determine if something is contaminated or something's in some solution or something, and it uses electrical impedance. It's more like a proximity sensor. If something disturbs as it passes through the tube here, let's assume this is a tube, something's passing through here, it disturbs that magnetic field, and you'll see it turn up as changes in voltages. You disturb magnetic influence is done by charging up coils, and if those are disrupted, it may take more voltage in order to maintain a particular magnetic. Well, we don't have to go through all those things. The idea is we can measure it by looking at the impedance or a change in that magnetic field. And it's really easy to do, and you can have it print to a printer so it counts it. Here's flow cytometer. You can have your sampling going in and the light source, and it can determine between different, you can put fluorescent stains on things, all sorts of stuff, and you can determine as the cell goes through, you get a pulse, and when it leaves, then it's the end of the pulse. So you can see that that was the pulse. So that's a positive pulse, so that was 1 and 2 and 3, and you can count. And if you have different fluorescent stains, let's say, you get different counts. It would know it gets a different signal, intensity or voltage or something. And you can set those things and parameters for what you want to look for. And just another representation of Coulter counters, and just counting through an orifice. This is not as accurate as what I just showed you. And here is real-time PCR, and all I'm showing you here, don't get confused, is just DNA heat it. That's the key. DNA, because of the phosphate backbone here, will separate. It shakes the DNA. If I could shake the monitor really violently, the two strands of DNA would come apart. Then you cool it a little bit without having the bigger strands get back together. You can actually put in little primers, and these are small segments of DNA that have been DNA synthesized, most likely, to match the region. And then this region in between where the arrows are pointing to, that's what gets amplified in PCR. So not only do we amplify DNA, but we amplify exactly the DNA we want. Now, the key to this is if the DNA, the primers stick to DNA that's there, then we know that whatever organism that we generated the primers will hybridize to that DNA. We know it was there because we can amplify the DNA. If it wasn't there or is not there, we can't amplify DNA because the primers are useless. What are they going to adhere to? The DNA, it just doesn't match. And so that's kind of the essence of what's going on there. Selectively, they call that DNA barcoding, but amplifying DNA is a key part of that. But we're not supposed to get into too much of that kind of genetic stuff, although it's very interesting. And you can interpret PCR results. They usually plot it, and the number of copies will show up with graphs with densities because there's more DNA. DNA at 260 nanometers, I guess it is. I forget what it is, but you can measure DNA will vibrate. It starts to vibrate and it gives a signal, so we'd see DNA. Whatever these colors represent, that DNA is present. Where down here, it's most likely not. And there you go. It's PCR, real-time PCR. Put the steps of bacterial growth in order. Death phase, no, I don't think. Lag, exponential, stationary. So, given the ones, we have a lag phase. Well, let's give you the answers here. B, which is lag. Then we go to exponential. Then we go to stationary, and then we go to death. Okay. Well, that is this is the rest of the story for chapter 6. Everything is together. We'll beginning next time, moving into chapters 9 and 12, and we'll see how that goes. But for now, everything, the practice questions and everything are out there for you, and I'll talk to you next time. Be safe and be well.